As yesterday, I have covered the basic local alignment search tool, how it works and what is the purpose to do it. So I have performed it on my computer, and I have suggested to all the participants to do it on your computer, and if face problem, then contact accordingly. And today's topic is uh, the transcriptome data analysis. As topic depicts the large-scale transcriptome analysis and data quality assessment, filtering of the data, steps and tools to analyze the transcriptome data. So in each sector, like medical science, in agriculture sector, biology, biochemistry, whatever there are stream of the science, you can see the transcriptome sequencing are frequently used, and it is basically used for analyzing the different sample and their gene expression level uh, in different tissue cell samples. So now we are going to discuss about the how do you use the transcriptome data to analyze, and how can we generate the expression level for particular gene in different samples of the transcriptome. So now I am going to start. So what is RNA-Seq data analysis? So actually, RNA-Seq data analysis is a sequencing of RNA, basically mRNA, has long been recognized as an efficient method for gene expression analysis and remain the gold standard for the annotation of the genes. So compared with earlier methods, massively parallel sequencing of RNA has vastly increased the throughput of RNA sequencing and allowed global measurement of the transcript abundance. RNA-seq is essentially the sequence of RNA molecule from either a specific cell, tissue, or a species. And differential expression of the gene by comparing different sample is the main motivation for RNA sequencing. So before starting the RNA sequencing, these are some prior questions you should aware about this, that what method of RNA purification and you are using. Actually, it is part of the wet lab. So you must aware what method you are using and what read depth you will need to cover your transcriptome. Read depth, actually it is the sequencing depth you want. Actually for a higher accuracy you need a higher read depth by the sequencer. So which platform you are using for sequencing, and is there a reference genome available or not available, and if it is available, then your analysis will follow the reference based analysis. Otherwise you will go for the de novo genome transcriptome analysis. And how are you assessing the quality of your RNA? And again, question is how I got enough biological and technical replicates. So what are the biological and technical replicates? I will discuss in my next slide. And what is your motto to analyze your sequencing data in a single read manner or a paired read manner? And what is the read length you are preferring for analysis? So these are the basic questions before you are going to sequence. So I will discuss all these in my next slide. So first of all, you should know when you are going for genome, and that is DNA sequencing. So there are three main steps which are widely used for genome and DNA sequencing. That is genome assembly and SNP calculation and DNA methylation analysis. So by DNA or genome sequencing, you can generate the data according to these three steps. And for RNA sequencing, there are three different steps, like transcriptome assembly and gene expression level, particular gene expression and differential gene expression among the samples or tissue. So we are focusing on the RNA sequencing. So in RNA sequencing workflow, it is the general workflow for the RNA sequencing. So first step is the library preparation. In library preparation, you should go for the extraction of RNA, which contain poly A tails, and then you should fragment this RNA and convert it into cDNA, and after that ligate the adapters, and after ligating the adapters, you should go for sequencing, and sequencing provide you the read in the FASTQ format, and read will use for the further bioinformatics analysis. So what are the replicates? Actually, in RNA-Seq analysis, we should aware about technical and biological replicates. So in general, we are taking at least 3 number of technical and biological replicate for particular sample. So when we talk about the technical replicate, so from same tissue or species, we can select 3 different replicate samples and use them as a technical replicate. And for biological replicate, we use three different sample from same tissue, but from a different organism. So it is known as biological replicates. So condition are same, but tissues are different. Like in this case, you can see this these are the samples, and the library preparation and sequencing. It is for biological replicate 1 and it is for biological replicate 2. So different biological samples and same conditions. So measuring biological variation among those samples. So it is basically do for if we want if we have some different some same species plant like Arabidopsis thaliana, and we want to check which which is which condition will affect the Arabidopsis thaliana. So we can take the sample from one from in general condition, that is grown in the field condition, and one in drought condition. So we can select the sample, and now we can check their expression level, which gene are highly expressed in drought condition as well as in general condition. So for that purpose, we can use the RNA-Seq data analysis. So in RNA-Seq data analysis, these are some important terms we should know for sequencing. So these are the read. A read is a single sequence produced by a sequencer. It is in the form of FASTQ format. What is FASTQ format? I will show you in next slide. Another is a library. Library is a collection of DNA fragments that have been prepared for sequencing. So this is generally taking about individual sample, and flow cell, a chip of DNA, a chip on which the DNA is loaded, and that provided to the sequencer. So it is known as flow cell. And in flow cell, there is a lane, one portion of the flow cell, usually used for technical replicates or different sample. And then run. Run is an entire sequencing reaction from start to finish. So these are the main steps to RNA-Seq analysis. And because there are a lot of RNA present in organism, so we are focusing on the mRNA. So what are the basic steps? First one is the library preparation. In library preparation, we have used the mRNA, select mRNA, and then fragment it. And after fragmentation, we have to reverse transcribe it using and convert it into the cDNA for their stability, because we use the cDNA for stability reason, and remove the RNA strand. So in this way, we can synthesize the double stranded cDNA, and then we can ligate the adapters to the cDNA, then select the size selection and go for PCR amplification. So these are the some wet lab experiment uh, they are performed, and in after the preparation of the sample, we are going to do it for sequencing. So in sequencing, we can go for single end sequencing as well as paired end sequencing. So basically, paired end sequencing are used for in general purpose because it provide sufficient information. So this is a workflow, how we design the experiment and prepare sample, and how go for bioinformatics analysis. So these are the samples and total RNA extraction, and after that we can select mRNA and purify it and then fragment it, and after fragmentation we can go for a reverse transcription of into cDNA, and after cDNA conversion, we can adapt adapter ligation. Adapter ligation. After that, we can go for amplification, PCR, and after PCR, we have gone through the high throughput sequencing. It is image of sequencer, and sequencer will provide the raw data in the form of FASTQ format, and this data we will use in our computer and extract the information of differentially expressed gene, as well as what is the expression of particular gene. That will provide the biological insight. So suppose it is an example. You can see that sample 1 and sample 2, we are going to sequence these samples. After sampling, we have got the reads. These are the reads, and this is the gene A and gene B of sample 1. This is gene A and gene B of sample 2. So you can see that gene A of sample 1 has total 30 reads mapped at particular location, and in gene B, it is 10 read map at particular gene B location. And in same way, in sample 2, gene A belongs to 10 reads, and gene B belongs to 5 read. So if we calculate the differential, then we can find gene A in the sample 1 has 3 by 10 is equal to threefold increase. So the expression of gene A in sample 1 has a threefold change increase compared to sample 2. That's similar way the gene B is expressed twofold change compared to the sample 2. So sequencing, when we are going to sequencing, so we have to select a single end sequence or paired sequence. So single end sequence only one sequence, and in paired end sequence it is sequence 1.fastq and sequence 2.fastq. So these are the sequence of reads which generate from sequencer, and these are the some file format which are essential to know before analysis. So sequence format are the FASTA and FASTQ format, and feature format, that is GFF and GTF format, and alignment format, that is SAM and BAM format. So these all are the file format which required for analysis of transcriptome data. As you know, in my previous discussion, that what is FASTA format. So it is start from greater than symbol, simple format. However, in general, header line start with greater than followed directly by ID. Files can be fairly largely, even it is a whole genome, any residue type DNA, RNA, protein, but simple alphabet. So it is easy to understand about FASTA format, and you can see it is a short sequence, and it will be a whole genome sequence. So now we can understand what is FASTQ format. Actually, FASTQ format is likely FASTA format, but it has the property of each residue, each nucleotide. So it is coded in this form. So DNA sequence with quality metadata. So this score generate quality for particular nucleotide. So the header line start with @ followed directly by ID of the sequences, and then after the description. It may be raw data straight from the sequencing or processed data, trimmed data. So there may be some variation like Sanger sequencing, Illumina sequencing. To particular sequencer have some variation. So it can hold hundreds of million of records. It can be very large also, like 100 GB of data in FASTQ format. So what are the feature format? These are the GTF and GFF file, SAM BAM file, and BED file. So what is the GTF? GTF is a gene transfer file, and this is the structure of GTF file. It has the information of chromosome and source of the sequence. What are the gene feature like CDS, start codon, stop codon, and start location and end location. Then it is strand positive or negative. So these are the information stored in the GTF file formation. And what is the GFF3? The GFF3 is a general feature format that is tab delimited file to store genomic features, example, genomic intervals of the genes and gene structure. Basically, it contains the gene structure like in gene, what are the location of the gene and what are the 3-UTR exon region, intron region, 3-UTR, etc. So it is similar to GTF file, but it has some structural variation. So after that, you should know what is alignment. So a way of arranging the sequences of DNA, RNA, or protein to identify the regions of similarity that may be consequences of function, structure, or evolutionary relationship between the sequences. So how can we store this information about millions of reads that align to our reference genome? So in this way, the alignment occurs like suppose this is our genome sequence and this is a read sequence. So our read aligned to this genome in this way. So these are the similar nucleotide residue. So and these are the it is you can see it has a gap. So there are 3 gaps inserted, and after that these are the similar nucleotide residue. So this is the alignment file, that is SAM. So it is known as sequence alignment or map format. SAM file format stores alignment information. It is a plain text, contain quality information, metadata, alignment information, sequences, etc. So files can be very large, up to 100 or 200 GB or more. Normally converted to BAM to save the spaces, and this BAM file also alignment file, and you can see it is SAM file format. It contains the alignment information of particular read to any genome or reference. So BAM is also it is compressed form of the SAM file. Actually, it is store the information of SAM file, but it is compressed, but not human readable file format. It is just read only by computer, and it is created to short the size of the SAM file. It is approx 1/5 of the SAM file, but it's still it may be a large like 10 GB, 20, 50 GB. So these are the basic file formats which are necessary to know before analysis. It is a simple workflow which is generally used to analyze the transcriptome data. First, experiment design. Second, library performance and laboratory performance. Sorry, third one is the data analysis. So in experimental design, you should know what library type you are preparing, sequencing depth, how much sequencing depth you are required, and number of replicates, how much replicates you are required. So this is experiment design. And laboratory preparation. In laboratory preparation, you follow the RNA extraction as well as library preparation, then sequencing by sequencer. After sequencing, the bioinformatics analysis of the data generated from the sequencer. So This is the first step, that is quality control analysis. And after that, the read were mapped to the reference genome. And after that, quantification. Quantification means how much genes have mapped read. And after that information, we can go for the differential analysis. So it is elaborated form of the experiment. Suppose you have two samples, 1 and 2. Then you go for experiment design and RNA extraction and purification and library preparation. And library preparation, you can go for the sequencing. After sequencing, you will generate raw read 1 and read 2, that is for parent data. And these read 1 and 2 will use for quality analysis, that is fast QC. It is a tool used for quality analysis of this raw data. After that quality analysis, we can go for read alignment. If we have a reference genome, then we can go for the reference based analysis. Otherwise, we can go for the de novo genome, transcriptome analysis. We will discuss the what are the tools required for reference based transcriptome data analysis, as well as de novo without reference based transcriptome data analysis. And after that, we can go for quantification. Quantification, it will be gene level or transcript level. And after quantification, we can go for differential expression testing, and that will generate in the form of visualization graph, visualized in graph and tables, and you can find out the biological insights of your experiment. So this is the first input data for analysis, like control. These are the replicates, and these end 1 and 2. So these are the two, because these are all the paired end sequences. So R1 and R2, and control R1, control 2 R1 R2, and these are the treated condition, knockdown R1 R2, knockdown 2 R1 R2. So these are the samples in reads generated in FASTQ format that will use like this flow chart. So first of all, we can go for the quality check of our generated reads by FastQC tool. And if it has some information like like our read have adapter data, and our read have some improper data, so we can use this FastP tool. FastP tool used for the quality control and filtering of the data. So we can filter our data according to our quality score. And what quality score I will discuss in my next slide. And after that, we can go for the mapping of our quality filtered data by HiSet2 tool, that is used for indexing and mapping to the reference genome. And after that, we have used this feature count tool, that is generate the read count data, that is abundance calculate. And this abundance calculated matrix will use by DE-Seq tool, that is an R package used for differentially expressed transcript or gene. After that, we can go for downstream analysis of annotation of our transcript or genes. So quality control, why quality control? Because it is essential for downstream analysis. Then decided sensibly on which data can be filtered out from the downstream analysis. And you might find yourself going back to that step several times during downstream analysis. So what are the quality score? So in general, you can see that this green area, if you go for the first queue of your raw read analysis, then you can find these type of graphs. So if all the reads are fall in this green area, it means the quality of your data is good. And if it appear something like this, then your read quality data is low quality. So before an analysis, you have to know your data is quality or quality data or not. After that, you can go for further. So it is score. Generally, we use the quality 30 data for our analysis. So if our quality data is not good, then we can use the fast P tool. It is an ultra fast all-in-one fast Q pre-processor tool. It will generate the data and trim the reads which are low quality reads. So you can see this is a result we have obtained by running fast P. So it describes how much data before filtering, that is 15.7 gigabyte data that have Q30 bases, and total our data sample 1, sample 2, sample 3, and sample 4, and what are the GC content. So these these are the after filtering data. Some of them data have bad reads. So then first we remove this bad read and filter the adapters and will generate the data. So it is 10.7 gigabyte data, and in case of this 6.4, 14.2, and likewise. So so if we have a reference genome, then we use this bioinformatics pipeline for our sequence analysis. So bioinformatics pipeline we have designed for reference-based transcriptome analysis. So before analysis, we have gone for the quality check and filtering. So this is the command you can see, fast QC, and then sample 1 and sample 2 name. These are the replicate samples. So likewise we have to do it for all the read generated for our sequencing results. So first of all, we have gone for by the fast QC, and if our fast QC report have some poor quality data, then we have to use fast P tool. And fast P tool, this command will use for fast P analysis, like fast P and minus I hyphen I sample R1 dot fast Q dot dz, capital I sample R2 fast Q dot dz, and after trimming and filtering, these two reads were generated, like trimmed R1 fastq.dz and trimmed R2 fastq.dz. So these are the reads which have some low quality reads, and after using this fast P, this will generate the quality filtered data in the form of trimmed R1 and trimmed R2. So now we have the trimmed data. Now we can use this trimmed data for our mapping purpose. So for mapping analysis, we have gone with the HiSet tool. Actually, there are many tools for mapping, like star, HiSet, string tie. In our case, we are using HiSet tool. So first of all, we have to build the index of the genome data which we are using as a reference. So here is the name of that genome, like Arabidopsis thaliana genome in FASTA format. Whatever you have genome, you can put it here, name here, and this will generate the indexing for that genome. And once indexing generated, then we can use for it for mapping. So this command will use for mapping of our data, HiSet2 hyphen X and genome. Here genome is the name of this. Actually, this index is used for our analysis. So here genome means index data of that particular reference. So one here one is sample 1 dot fastq. It is actually this one, trimmed R1 fastq.dz, and sample 2 is the trimmed R2 fastq.dz, and hyphen S will generate the output in the alignment format, that is SAM format. So in this way we have obtained the output in the SAM format. So after getting the output in the SAM format, we have used the SAM tool. So SAM tool is basically used for the conversion of SAM to BAM and index the BAM file, sort and index the BAM file for our further downstream analysis. So once we got the SAM file of alignment, then we have go for uh, aligned with sorry, we have to convert this SAM into BAM. So this is the command SAM tools view -s -b and align that SAM. This is output file. Align.sam will convert in the align.bam. So once we get this BAM file, then we can use this SAM tool again to sort this BAM file according to the coordinates. This will sort this aligned.bam form file into the aligned sorted.bam file. And once we got the sorted BAM, then we can index with the SAM tool, like SAM tools index aligned underscore sorted BAM. Once we have generated the indexed file, then we have go with the feature count, that is abundance analysis. So feature count is a tool that is used for the abundance estimation. Like it calculate how much gene have mapped read, number of mapped read. So it will generate the result in the form of count. Suppose one gene have mapped with 10 reads, then it will count, it will generate that gene 1 has 10 count. So it likewise it will generate a matrix of particular gene with mapped read. So this is whole command. For that analysis, we have to one file, that is annotation.gtf. So once we have the genome file, so in the biological databases like NCBI and Ensemble plant, there are GTF file also available apart from the genome.fasta. So we have this genome file as well as annotation.gtf file. If we have these two files, then go for feature count analysis. So it will generate the count number of counts for particular reads. So this will you can see this is the mapping stat. So when you got the output in SAM format, so it will generate the mapping statistics for particular sample. So you can see that sample 1, 2, and 3, which are this condition, which have this condition. Sample 4, 5, 6, which have this condition. So likewise, there are a number of map reads found for each particular sample. So total uniquely map read obtained for this sample is 79.59%. And in case of sample 2, it is 64. So basically, if we have a higher number of map reads, then our analysis will provide a good result. So approx more than 80 to 90% we have. If we have the map read, then it is good for our analysis. And if it is lower percentage, then we should go back, and again we have to sequence the sample and go for mapping analysis. So after getting the feature count matrix, like count data matrix, we have used the differential expression analysis tool. So these are the differential expression analysis tool, and these two tools are widely used for differential expression analysis, that is DE-Seq2 as well as HR. So in our case, we are using DE-Seq2 for differential analysis, expression analysis. So these are the command actually which are used for the differential expression analysis. So first of all, we have installed our computer with the RStudio. RStudio is a freely available tool. You can install it on your computer. So once you have to install this R Studio, okay. Once you have to install this R Studio, um, this will look like this. So you can see it is actually a console region. Here you can type your command. Like in our case, we have used this library for our analysis. So just type this command and enter. So it will load the DESEC2 library for our analysis. Once this library is loaded, then we can go for DESEC2 analysis for our data. So for data, we can import the data from here directly to our studio. Like you can see from there you can upload the data of read that actually it is data in the form of read table. So you can load your data in this way. Once you have upload the file from here, you can type the command and you just put the file name here. And this file actually have the information of read count. What is which file is generated by the feature count method, like gene ID, sample 1, sample 2, sample 3, whatever. Okay, gene 1, 2, 3, 4. And this is the read count information. Like gene 1 have 23 reads mapped, and in sample 2 it is 24, 34. It is in sample 3. It is total read count is 45, and likewise you can see. Okay, so this is actually read count matrix. You can it actually it already generated by using feature count method, because feature count method and all the tools are actually installed and run on Linux operating based system. So we just provide you demo how the data looking like and how can we upload. So this is count matrix, and it is in the tab delimited format. It is saved in the on the desktop. So we just upload by this way count matrix. Here is the heading, and first one line, our first line use first column as the row name. Now upload this. You can see the data is uploaded. Now we can provide the file name like count matrix. So now the data is stored in the form of count data. So likewise, we have another file that is known as metadata format. What is metadata format? Actually, for our DE-sec analysis, we have prepared one file also for our metadata analysis. That is, in this file, we have to provide the sample information in this form. Sorry, it has not. It have no information of gene. Actually, it have information of sample and their condition. So this is our metadata file. We can store it in this way. This is also in the tab delimited format. So it provide the tool the information of which sample have the control condition and which sample have treated condition. So likewise, we have to upload this file also in the DSEC in the RStudio tool first column. So you can see that this file has been uploaded in our console in our RStudio, and now we have to go for this. We will provide the column data file name, that is metadata file name, metadata. So now we have to upload these files which are generated. First one is generated by feature count method, and second one we have designed this file. After that, we have to check that all the columns present in our feature count data matrix are also present in the data column. So these two commands will check it. If this command will find true, then we can go for further. Otherwise, we have to check our data and metadata. After that, we have set the condition to factor. Actually, so there are some characters also in our files, so it will generate a factor, and after ensuring the condition is a factor, after that create DE-sec to data set. So this command will generate the DE-sec dataset. Like DDS is a command, and will use in this command we have to use the count data, that is feature count matrix, as well as our column data as metadata I have designed. And in this design, we have provide the condition. Actually, we will tell this software what conditions are available in our metadata, that is control condition and treated condition. So it will identify the conditions accordingly. So now we set the reference condition. So in our case, we have to set the reference condition as a control. So it will take as a reference as a control, then it will calculate the treated sample in reference of the control. control data. And then now run this command, and these are the some command. After running this command, we have to check the summary of result. So summary of result will provide this information, like out of 71,425 genes at adjusted p-value greater than equal to 0.1, total LFC log fold chain is greater than 0. So these are actually known as upregulated genes. And in our case, it is 27%, 19,482. And if we talk about the down-regulated genes, that is a log fold chain have less than 0, then it will 30%. That is 21,330. So likewise, we have saved that result in that .csv file that will open in the Excel, and after that we have to use the data according to our cutoff. In general, we are using data for further analysis. The p-value we should use the 0.05, and log fold chain is approx 2, minimum 2 log fold chain value we have to use. So this is the total analysis by reference based analysis. And if we talk about the if reference is not available, then we have go for the de novo transcriptome assembly and analysis. So you can see, if you have not the reference sequence, then these are the requirements, like deep sequencing and longer reads and thorough quality controls and large memory and multiple processors for this analysis. These are the some basic tools which are used for the de novo transcriptome assembly, and among them Trinity is most widely used software for de novo transcriptome analysis. After differential analysis, we can go for normalization of our data. Actually, why we need normalization? Actually, more reads map to the transcript. If it is long, if one transcript or gene is gene length is very long, then many number of reads will mapped, and if in same way in the gene which have short length, the map read are less. So at higher depth cover, the data we have use is high at higher depth coverage. So if you have the higher depth coverage, the number of reads also increases. So that's why we have there is a need for normalization of our data. So it will actually features of different lengths. Actually, it will generate the data to compare between the samples, and it make easy to compare basically. So there are some methods for normalizing our data. So first, this one is the fragment per kilobase of the transcript or per million mapped read, FPKM. So FPKM is simple expression level normalization method. The FPKM normalizes read count based on the gene length and the total number of mapped reads. So the total number of mapped read information we have obtained from SAM files, and gene length we have obtained from GTF file or GFF file. So in this way, we have to calculate the FPKM value. So FPKM formula is the number of reads mapped to the particular gene and into 10 to the power 9 divided by total number of mapped reads in the experiment and length of the gene in base pairs. Suppose we have read map to a particular gene is 5,000, and total mapped read for particular experiment is 20 crore, and gene length is 2000. So 2000 base pair. So we can calculate the FPKM by using this formula: 5000 into 10 to the power 9 divided by 2 crore, 20 crores into 2000, that is 125 for particular gene. So likewise, we have to calculate it for all genes in Excel. You can use the formula, and that's way we can compare the samples in the normalized form. So if we not use this FPKM, then we have to calculate. We have to use like if we have a gene that mapped read are 5000 and one gene have only 20 reads mapped, then it is not easy to compare such condition. That's why we use the normalized data. So statistical results actually a list of significantly differentially expressed gene. So here why we use the significantly, actually significantly, when we use the significantly term, then we have to use the p-value and p-adjusted value and log fold chain value. So we can provide the cutoff, statistically cutoff for the data, and in general it is p-value, it is 0.05, and log fold chain value is 2. So if we have provide this value, then the data we obtained is significantly differentially expressed, and then we can use the differentially expressed gene for heat map analysis, Venn diagram, and more for annotation. So you can see these are the symbol of genes, and this is log fold chain value. It is in minus, it means these are the down regulated genes, and the p-value, and the FDR, it is adjusted p-value, and these are the counts for particular genes. So these are the counts, and likewise you can see this is the heat map which is generated like in blue color. It is showing that -1, and in red color it is +1. So minus means these are the genes that are differentially expressed, but these are negatively differentially expressed, and these are the upregulated genes which are shows in red color. So this is the actually differential gene expression analysis result. And by this way you can see that in your sample, like whatever your experiment, like your experiment have some drought condition, some in general condition, some specific disease condition. So you can compare which genes are highly expressed and which gene are positively means upregulated, and which genes are down regulated in your samples, and you can go for biological insights accordingly. So after that analysis, you can use some gene ontology websites which are designed to analyze the genes for their gene ontology analysis, like pathway enrichment analysis, and what are the biological process. You can select the species from here. So there are many databases. From there you can annotate your data. So final thoughts and stay observations are the one is the think carefully about what your experimental goals and are before designing your experiment and choosing your bioinformatics tools. So as we have discussed that if you have the reference genome of your experimental data, like you are working on Arabidopsis thaliana or rice or wheat, so there are availability of reference genome, like whole genome, annotated genome is available in the ensemble plant TAIR database, and NCBI database. There are availability of whole gene annotated genome in the FASTA format, as well as GTF and GFF file also available. So if you have designed your experiment as in some particular plants which are Arabidopsis, rice, maize, wheat, so there are available reference genome. And if you have some novel plant analysis for analysis of novel plants, so if in that condition there is not availability of reference genome, then you can go for de novo analysis. And de novo analysis, we have suggested the tool like Trinity and Velvet. So you can go for the de novo analysis by using that tool. And the second one is when the doubt, if you have doubt for analysis, then search on Google, and these are the sites like BioStars, Bioinformatics experts are available here. You can ask them question and they will answer whatever your query. And seek answer the next generation sequencing community. Here also you can post your query, and the experts will answer your query. Another good resource, if you are not ready to use the command line routinely, then it is a Galaxy web server. Galaxy NGS data analysis web server is available. You can just go there and make your ID. And once you have created your ID in Galaxy data, you can use all the tools which I have discussed here in our slide. You can use all the tools freely, and these are the actually graphical user interface uh, website. You cannot run any command. You just click on each tool and run the data analysis. So if you are new or not aware about the Linux operating system, how it is work, and what are the tools, how the command will generate the result, then you can use this Galaxy software. But the workflow is same, like fast QC, fast P, and filtering. After filtering, you can go for mapping. For mapping, you can use HiSat2, and after mapping, you can use the feature count. So all the tools are available in this website. You can go and upload your data and go for analysis. It is a web-based bioinformatics portal that can be locally installed if you have the necessary computational infrastructure. So you can use with your basic laptop or your computer. So these are the online resources we have, as we have discussed. So these are the actually basic slides where basically you can use the transcriptome data analysis of your samples. So I can show you the Galaxy. It is a web server. Here you can analyze your data by uploading your raw data, which is generated by sequencer that company will provide you. If you have not your sequencer at your end, then you have go for the company for sequence data analysis, and company will provide you the raw reads, and you can upload the raw reads in this. First of all, you have to create your login ID and password, and once you have login, then you can upload the data in this way. You can upload here, select your raw read files R1, R2 for your samples, all samples. So once you have upload the data, this will actually store here. And after that, you can use the tools like fast QC. Suppose this one is fast QC. So so here you can directly upload the raw reads here and just run this tool, and this will generate the quality of your data. If you have bad quality data, some reads are actually have some score which is not acceptable for RNA-Seq analysis, like Q30 is less than 30, then you can go for FastP. Here is the FastP tool. You can upload your data directly here, raw reads, and it run this tool, and it will generate the data in the form of trimmed data, which is quality filtered data. After that, when you obtain the quality filtered data, and then you have to go for mapping analysis. HiSat2. HiSat2, this is a tool which is used for genome reference based analysis. So first you have to upload your genome, and here is the name of the genome, and if you select your data analysis, that is paired in data or single in data. So in our case it is paired in. So in paired in, you can upload FastQ file, one file, and FastQ2 file for a particular sample, and after that you can run this tool on default basis. That will provide you the information, mapping information, in the form of SAM and BAM. If you want to convert the SAM file, then there is a tool, SAM tools. So SAM tool view. So this command will convert your SAM file into BAM file. Just run this. After that, SAM tool sort. SAM tool sort will sort your BAM file and then index. SAM tool index. Okay, SAM tool in when you sort the BAM file, then use these tools, SAM tool index, sorted BAM, and yes, this will use for the index, and after that you can use the feature count. Feature count. This tool, feature count, will generate the count reads per particular gene, and in this you can use the GFF file for your uh, reference, and then run it directly, and you will obtain the read count file. So from here, FastQC to gene count, these all tools are freely available on this Galaxy web server. You can use directly without any command or etc. So after getting this read count matrix, you have to use this RStudio software for your analysis, and after using this, you can find the expression analysis data. Suppose you can plot this particular graph in your RStudio data. R Studio. Here you can see that these are the log two-fold chain. Log two-fold chain it is so that these are the red color. So these are the genes which shown in the red color. So these are the above than 0. So you can see that these are the upregulated genes, and if it is less than 0 in the minus, so these are the down-regulated genes. So in this way, you can plot your data, and likewise you can plot your differential expression data also, and your annotation. So this is all about general bioinformatics analysis of your RNA-Seq data, and you can do it by yourself. You can just download the transcriptome data, which is available on NCBI website. You can use that data for your training purpose, and use by Galaxy, as well as the software you can locally install on your computer. So this is all about basic about the transcriptome data analysis. And if you have any query, then you can